Vitamins are organic compounds. They are essential micronutrients that the human body needs in small quantities for the proper functioning of its metabolism. Vitamins cannot be synthesized by the human body, either not at all or not in sufficient quantities, and therefore must be obtained through the diet.
Vitamins are classified as either water-soluble or fat-soluble. Water-soluble vitamins can be further divided into B vitamins and vitamin C. Today, we will cover these three groups of vitamins. First, we will look at B vitamins. B vitamins, or their active forms, have nitrogen-containing heterocyclic rings. Except in biotin, nitrogen-containing rings are aromatic. First B vitamin that we are going to cover today is thymine, vitamin B1. This is thymine. Thymine has two nitrogen-containing rings. Active form of thymine is thymine pyrophosphate, or TPP. This is TPP with two phosphate groups. Thymine is a coenzyme for three dehydrogenases in oxidative decarboxylation of alpha-keto acids. More specifically, it is a coenzyme for E1 in pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase, and branch-chain alpha-keto acid dehydrogenase complexes. For example, pyruvate is decarboxylated by E1 to form a hydroxyethyl derivative bound to the reactive carbon of TPP. For details, please refer to lecture Glycolysis and pTH in this lecture series. Thymine is also a coenzyme for transketolase in the HMP pathway. B1 is enriched in whole grain cereals, meats, legumes, and nuts. Here is clinical correlation. Thymine deficiency is most common in alcoholics and in countries where polished or white rice forms a major part of diet. General feature of thymine deficiency is decreased ATP production. Since thymine deficiency impairs energy metabolism, intravenous glucose infusion may precipitate acute thymine deficiency in alcoholics. Tissues that are most sensitive to thymine deficiency are muscles and nerves. When symptoms manifest in cardiovascular system, such as dilated cardiomyopathy, tachycardia, vomiting, convulsion, it is called wet beriberi. When symptoms manifest in the nervous system, such as polyneuropathy, difficulty in walking and speech, loss of tendon reflexes, and involuntary eye movement, it is called dry beriberi. When ocular disturbance, ataxia, and loss of memory occur, it is called Wernicke-Korsakoff syndrome. All these are caused by thymine deficiency, secondary to alcohol abuse. The second B vitamin is riboflavin, vitamin B2. This is riboflavin. It consists of two components, pentose alcohol and the flavin base. Active forms of riboflavin are flavin mononucleotide, or FMN, and flavin adenine dinucleotide, or FAD. FMN is derived from riboflavin by phosphorylation at 5' position. FAD is derived from FMM by addition of adenosine monophosphate. FAD is a coenzyme of succinate dehydrogenase and 3-alpha keto acid dehydrogenase complexes that I had described to you earlier. FMN is a component of complex 1 in electron transport chain and many flavoproteins. Riboflavin is enriched in milk, eggs, meat, and green leafy vegetables. Please note, riboflavin and its active forms are UV sensitive and they can be destroyed by UV. FAD can be reduced at positions 1 and 5 in the flavin base and become FADH2. FADH2 is a high-energy compound. Here is clinical correlation. Patients with riboflavin deficiency have dermatitis, cheilosis, and glossitis. The third B vitamin is niacin, vitamin B3. This is niacin. It is also known as nicotinic acid. The active forms of niacin are nicotinamide adenine dinucleotide or NAD and NADP. NAD is derived from niacin through a multi-step pathway. NADP is derived from NAD by phosphorylation at 2' position in a ribose. Excess tryptophan is metabolized to niacin and supplies about 10% of recommended dietary allowance. Niacin is enriched in meat, whole grain cereals, tryptophan-containing foods. Niacin is a coenzyme of many enzymes in redox reactions involving hydride ion transfer. This is hydride ion. NAD is reduced to NADH by accepting a hydride ion. Here is a clinical correlation. Niacin deficiency causes pellagra, described by 4 Ds: diarrhea, dermatitis, dementia, and death. The fourth B vitamin is pantothenic acid, vitamin B5. This is pantothenic acid. It consists of 2 components, beta-alanine and pentoic acid. The active form of vitamin B5 is coenzyme A. This is coenzyme A. It consists of 3 components, cysteine, pantothenic acid, and adenosine diphosphate. It is derived from pantothenic acid and cysteine in a 5-step process that requires 4 ADP molecules. Coenzyme A is a coenzyme of fatty acid synthase complex, alpha keto acid dehydrogenase complexes, and many more enzymes. In particular, the thia group from the cysteine is actively involved in reactions. Vitamin B5 is found in a wide variety of foods. As a result, vitamin B5 deficiency is very uncommon. Vitamin B6 refers to a group of chemically similar compounds which can be interconverted in biological systems. Pyridoxine, pyridoxal, and pyridoxamine. The active form of vitamin B6 is pyridoxal phosphate. Vitamin B6 is enriched in whole grain cereals, eggs, meat, fish, soybeans, and nuts. Isoniazid, a drug used to treat TB, has a structure very similar to pyridoxine. Isoniazid may induce vitamin B6 deficiency. On the other hand, excess uptake generates toxicity. Important reactions that require PLP can be divided into 4 classes. First classes of reaction involve an amino group, such as transamination by ALT and AST, deamination by histaminase. Second class of reactions involve a thiol group, such as transsulfuration. It is transfer of thiol group from homocysteine to serine, catalyzed by cystothionein beta synthase. Cleavage of sulfur gamma carbon bond, catalyzed by gamma cystothioneinase. Third class of reactions involve a carboxyl group. For example, decarboxylation by aromatic amino acid decarboxylase, histidine decarboxylase, phosphatidylserine decarboxylase, and ALA synthase. Fourth class of reaction involve cleavage of glycosidic bonds. A well-known example is glycogen phosphorylase. Here's clinical correlation. Patients with vitamin B6 deficiency have seborrheic dermatitis-like eruption, atrophic glossitis with ulceration, angular cheilitis, conjunctivitis, intertrigo, along with neurologic symptoms, including peripheral neuropathy. Vitamin B6 deficiencies are seen in alcoholics, infants given deficient formula, and women on oral contraceptives. Isoniazid, used to treat TB, may induce vitamin B6 deficiency by 2 mechanisms. First, isoniazid metabolites directly attach to and inactivate pyridoxine species. Second, isoniazid inhibits the enzyme pyridoxine phosphokinase. This enzyme is required to activate pyridoxine to PLP. Biotin is also known as vitamin B7. This is biotin. It is a coenzyme in carboxylation reactions. And here is the site for carbon dioxide attachment. Biotin is produced by bacteria in the intestine. Patients with biotin deficiency have alopecia and seborrheic dermatitis. Biotin deficiency may be caused by eating raw egg whites, which contains avidin, and use of broad-spectrum antibiotics. Cobalamin is known as vitamin B12. 2 enzymes require cobalamin. One is methionine synthase. It is also known as homocysteine methyltransferase. It catalyzes synthesis of methionine from homocysteine. Second enzyme is methylmalonyl-CoA mutase. It catalyzes isomerization of methylmalonyl-CoA derived from oxidation of fatty acids with odd number of carbon atoms. Cobalamin is found in meat, dairy products, and eggs. It is also found in seaweed and mushroom in a small quantity. It is not present in plants. Active forms of cobalamin are methylcobalamin and deoxyadenosylcobalamin. Here is cobalamin. It contains a choline ring and a cobalt. A choline ring is made up of 4 pyro rings. 2 of the pyro rings are linked directly, rather than through a methane bridge. Cobalt is held in the center of the choline ring by 4 coordinate bonds with 4 nitrogen atoms from the pyro groups. When methyl group is attached here, it is methylcobalamin. When 5'-deoxyadenosine group is attached here, it is 5'-deoxyadenosine cobalamin. Dietary vitamin B12 complexes with the protein R factor secreted in saliva. In the small intestine, pancreatic enzymes cleave R factor, which allow vitamin B12 to complex with intrinsic factor. Intrinsic factor is produced by parietal cells in the stomach. Vitamin B12 intrinsic factor complex is absorbed in terminal ileum. Intrinsic factor is necessary for absorption of B12 in terminal ileum. After absorption, vitamin B12 is bound to transcobalamin in plasma and delivered to cells that need it, or stored in the liver. Here is clinical correlation. Vitamin B12 deficiency causes accumulation of intermediate fatty acids, leading to neurological effects. Deficiency in either vitamin B12 or folic acid causes increase in plasma homocysteine levels, which damage blood vessels and pose a risk for thrombosis. Common causes of B12 deficiency include pure vegan diet, chronic pancreatitis, terminal ileum disease, and autoimmune diseases. In particular, pernicious anemia is caused by an autoimmune disease. It is caused by inability to absorb B12 because of autoimmune destruction of parietal cells. Pernicious anemia is a megaloblastic anemia. Treatment requires lifetime intramuscular injection of cyanocobalamin. Please note that therapy of megaloblastic anemia is often initiated with folic acid and vitamin B12 together until the cause of the anemia can be determined. This is due to the fact that folic acid can partially reverse hematologic abnormalities of vitamin B12 deficiency and can mask a cobalamin deficiency. The last B vitamin is folic acid. This is folic acid. When double bond between positions 7 and 8 is reduced, it becomes dihydrofolic acid. When double bond between positions 5 and 6 is further reduced, it becomes tetrahydrofolic acid. Active form of folic acid are tetrahydrofolate derivatives. They are important in one-carbon transfer reactions, especially in DNA synthesis. Folic acid is enriched in green leafy vegetables, liver, lima beans, and whole-grain cereals. Here are the clinical correlations. Folic acid deficiency is most common vitamin deficiency in the US, especially in pregnant women and alcoholics. It causes megaloblastic anemia. Folic acid deficiency in pregnant women can cause severe birth defects known as neural tube defects. Folic acid is ingested as polyglutamates. They are converted to monoglutamate in jejunum by intestinal conjugates. Here are some examples of monoglutamate. Two are highlighted. One is N10-formyl tetrahydrofolic acid, and the other one is N5-N10 methylene tetrahydrofolic acid. Monoglutamate is absorbed in jejunum. Bacteria can synthesize folic acid de novo. 7,8-dihydroxypyrophosphate pyrophosphate reacts with p-aminobenzoic acid to form 7A dihydroteroid. This reaction is catalyzed by dihydroteroid synthase, or DHPS. After conjugation with glutamate, it becomes dihydrofolic acid. Dihydrofolic acid is reduced to tetrahydrofolic acid by dihydrofolate reductase. Two groups of antibiotics block this pathway. DHPS is blocked by sulfonamides, and dihydrofolic reductase is blocked by trimethoprim. Folic acid deficiency can be caused by diet lacking fluid and vegetables. Chemotherapeutic drugs such as methotrexate inhibit dihydrofolic reductase. Phenytoin inhibits intestinal conjugates, whereby it inhibits absorption of folic acid. Absorption of folic acid can also be caused by oral contraceptives, alcohol, and celiac disease. Finally, rapid growing cancer cells compete with normal cells to consume folic acid. Here is a summary of B vitamins. This map can be divided into 4 regions. Region 1 on the top represents carbohydrates metabolism. Region 2 covers the center and the bottom. It represents TCA cycle and electron transport chain. Region 3 represents metabolism of amino acids. Region 4 represents fat metabolism. For example, degradation of glycogen requires PLP. Oxidation of glyceraldehyde 3-phosphate requires NAD. Carboxylation of pyruvate requires biotin. Using the same method, we can correlate each vitamin with enzymes in other pathways.
Now, let's move to part 2. We will look at vitamin C.
This is vitamin C. Vitamin C is a coenzyme in hydroxylation reactions on proline and lysine in collagen synthesis. Vitamin C is an antioxidant. It helps reduce free radicals. Vitamin C is found in citrus fruits, potatoes, tomatoes, and green vegetables. Here is a clinical correlation. Vitamin C deficiency causes scurvy. It is characterized by bruising, bleeding gums, weakness, fatigue, and rash. Causes of vitamin C deficiency include malnutrition in sailors, pirates, explorers, and soldiers, and loss of vitamin C due to pasteurization. Heat and storage also destroy vitamin C.
Now, let's move to part 3. We will look at fat-soluble vitamins.
Fat-soluble vitamins have several common features. They are found in fats and oils. They require bile for absorption. They enter the lymph before entering the blood. They are stored in fatty tissues, and they may reach toxic levels since they are not readily excreted. First fat-soluble vitamin that we are going to cover today is vitamin A. Vitamin A has 3 forms: retinol, the alcohol form, retinol, the aldehyde form, and retinoic acid, the acid form. They are derived from beta-carotene. This is beta-carotene, which is split to form retinol in intestine and liver. Beta-carotene is found in beef liver, sweet potatoes, and carrot. Beta-carotene is split to form retinol. Each retinol molecule reacts to give retinol. Retinol is esterified and packed into chiral microns and delivered to the whole body. In the target tissues, retinol is converted to either retinol in retina or retinoic acid in most other tissues. Retinoic acid binds retinoic acid receptors, which are nuclear receptors. Retinol, specifically 11-cis-retinol, is a component of the visual pigments of photoreceptors. It requires continuous replenishment. Vitamin A maintains epithelial tissue and skin. Especially retinoic acid controls expression of the keratin gene. Retinoic acid also plays a role in sperm development and fetal development. Vitamin A are therapeutic agents. For example, all-chain retinoic acid, also known as tretinoin, is used to treat psoriasis and promyelocytic leukemia. 13-cis-retinoic acid, also known as isotretinoin, is used to treat severe acne. Here is a clinical correlation. Vitamin A deficiency causes night blindness. It also causes acne and psoriasis. Causes of vitamin A deficiency include iron deficiency, excess alcohol consumption, and conditions associated with fat malabsorption. Here is in the nucleus. Retinoic acid receptor RAR heterodimerized with retinoic X receptor RXR. In the absence of ligand, this dimer binds hormone response element in the DNA, complex with co-repressor protein. No transcription occurs. Binding of retinoic acid to the RAR, resulting dissociation of co-repressor and recruitment of co-activator. Gene transcription begins. Second fat-soluble vitamin is vitamin D. Vitamin D is not an essential vitamin for most people, since the body can produce it. Synthesis of vitamin D involves three organs: skin, liver, and the kidney. Main function of vitamin D is to control calcium absorption in the intestine. Vitamin D is found in fatty fish, pork, liver, egg yolk, and mushroom. It is also found in fortified food. Vitamin D can be obtained by exposure to the sun, 10 to 15 minutes, several times a week. The precursor for vitamin D is 7-dehydrocholesterol. This is 7-dehydrocholesterol. It is an intermediate from cholesterol synthesis. Synthesis of vitamin D begins on the skin, where 7-dehydrocholesterol is converted to cholecalciferol or vitamin D3. In the liver, vitamin D3 is converted to 25-hydroxy-D3 by 25-hydroxylase. In the kidney, 25-hydroxy-D3 is converted to 1,25-dihydroxy-D3 by 1-alpha-hydroxylase. This is the active form of vitamin D. Vitamin D receptor is a member of the superfamily of steroid thyroid hormone receptors. Vitamin D receptor form a heterodimer with the retinoic X receptor RXR and bind to the hormone response elements on DNA. Upon activation by vitamin D receptor controls transcription of specific genes As an example, vitamin D receptor control transcription of transport proteins, which are involved in calcium absorption in the intestine. For example, vitamin D activates transcription of three genes coding for calcium binding proteins, calbindins, calcium pump, and calcium channel. Here is the enterocyte, and this is intestinal lumen. TRPV5 or V6 is a calcium channel that brings calcium into the enterocyte. Calbindin D is a calcium binding protein that moves calcium from one side of the cell to the other. Calcium pump PMCA pumps calcium out of the enterocyte. Here is a clinical correlation. Vitamin D deficiency causes rickets in children and osteomalacia in adults. Here are children with rickets. It is characterized by the continued formation of collagen matrix of the bone, but incomplete mineralization. Osteomalacia is loss of minerals, especially calcium, from the pre-existing bones. Causes of vitamin D deficiency include insufficient exposure to sunlight, renal failure, and hypoparathyroidism. Vitamin E is a group of vitamins that consist of 8 naturally occurring tocopherols. Alpha-tocopherol is the most active one. This is alpha-tocopherol. Vitamin E is an antioxidant. It reduces free radicals and prevents non-enzymatic oxidation of cell components by molecular oxygen and free radicals. Vitamin E is found in the liver, eggs, and vegetable oils. Vitamin E deficiency is almost entirely restricted to premature infants. In preemies, destruction of the red blood cells leads to hemolysis. Vitamin K includes two natural forms, vitamin K1 or phylloquinone and vitamin K2 or menaquinone. And here is K1 and these are K2. K1 is found in cabbage, cauliflower, chili pepper, and basil, among others. Vitamin K2 is made by bacteria in the gastrointestinal tract, absorbed and stored in liver. It meets half of the needs of this vitamin. Liver is enriched in vitamin K1 and K2. Vitamin K is a coenzyme of vitamin K-dependent carboxylase. It aids in blood clotting. And here is a clotting factor, and this is glutamate residue. It is carboxylated at the gamma carbon position, leading to gamma carboxylated glutamate or GLA residue. This reaction occurs in the liver ER and catalyzed by vitamin K-dependent carboxylase. Proteins that contain the GLA residues include factors 10, 9, 7, 2, protein C, and protein S. These proteins can be summarized in one mnemonic, 1972 CS. It represents factors 10, 9, 7, 2, protein C, and protein S. Here is clinical correlation. Vitamin K deficiency causes bleeding. Causes of vitamin K deficiency include malnutrition and use of antibiotics. Vitamin K deficiency can also be induced by anticoagulants such as warfarin and dicumarol. And here is dicumarol and this is warfarin. This is vitamin K. Dicumarol and warfarin are vitamin K analogs. Warfarin and dicumarol are competitive inhibitors of vitamin K epoxide reductase. This enzyme is required for recycle of vitamin K.